Okay. Just you two on here. Hopefully other people will join in as we go. At least it's recording. I'll just wait a little bit just to see if anything else comes. Yeah. I'm sure nobody else is coming. All right, let's go ahead and get started. You guys got here on time, so let's go ahead and do this. Oh, good. Okay. Waiting for her to connect. Good, good. Okay. So let's go ahead and get started with Unit 3. We'll start with microbial control, since that's what the first chapter was here. In Chapter 11, we were talking about the physical, chemical, and mechanical ways of removing microbes. So here, I mean, the physical and mechanical is going to be the same deal here, but if we're going to physically remove organisms and not by a chemical means, then we were using things like radiation, filtration, ultrasonic waves. I can't see it. Heat, whether it's moisture, dry, tindalization, that's in part of the heat. Cold desiccation, osmotic pressure. So, and then the chemical ones, we had all those, the halogens, including chlorines and iodines, the phenols, alcohols, which I misspelled. I apologize for that, but yeah, you know how to spell alcohol, hopefully. Hydrogen peroxide, surfactants, detergents, and quats, heavy metals, aldehydes, gases, dyes, acids, and alkalis. So that's kind of our list of everything that we had talked about in the chapter, of course, and we've separated it out now here, physical and mechanical versus the chemical. And if we look on the microbial control slide that they had given us, we'll go back to the other one soon. When you look at this, it kind of gives you that walkthrough of what's going on with all of those. So if you guys want to look through this, like I said when I was lecturing, this stuff is the stuff I would look over whenever you're trying to separate out not only like what is a physical agent, but as well as which ones can sterilize and which ones can't. If we're boiling water, if we're pasteurizing, those things, these right here, they're not going to sterilize. Boiling water doesn't sterilize, just normal hot water. Pasteurization, that tindalization, those are just exposures to heat and moist heat, but it's not going to be hot enough to actually sterilize it. We can remove a good portion of the microbes and make it consumable, but it will still have microbes in there. So that's important to remember that. And that's the same with UV radiation. UV radiation also, it says participants can now see your screen, but I was screen sharing that whole time. Hopefully that wasn't just now showing up. Anyways, UV radiation is non-ionizing, so that's going to be for disinfection as well. Whereas the other types of radiation, that ionizing radiation, X-rays and gamma rays and things like that, those can be used to actually sterilize, with enough exposure. So yeah, I would kind of go through these and try to remember which ones can sterilize and which ones are just going to disinfect. And then, like, if we talk about antisepsis, we're using that on like a living surface, like a body. And then their little terms are over here in this little box if you need to review those. I'd also try to understand a little bit about how these methods work, as far as like why incineration, why heat in general, let's just say that, why heat in general is effective at controlling microbes. And we had talked about, proteins and, basically breaking them down because they're denaturing them because of the heat. that's what's going on. That's why it works. That's why it kills things and gets rid of everything, because we can denature proteins. We can even, at some point, we get hot enough in an autoclave, which is something that involves steam and heat and pressure, to get rid of things, to sterilize things. We can even get rid of prions that way. So they normally can't be gotten rid of any other way, but we would have to pump it up to a much higher temperature than we would typically use an autoclave at. But we can get rid of them through heat and moisture by denaturing those proteins. And let's go back to this slide. Let's see, go through this. Right. Know the differences between sterilization, disinfection, antisepsis, and then decontamination and sanitation. Those are the same terms. But sterilization, meaning, removing all of the microbes. That's the only one that means, complete removal of all microbes. The rest of them are going to leave some microbes behind, typically endospores, right? That's a pretty common one that they'll leave behind. And so that's something to consider, and understanding like that antisepsis is dealing with the body surfaces, whereas disinfection isn't technically. So breaking those terms down into what they mean. So recognizing the difference between microbicidal and microbistatic. Again, we talked about this in the lab. That was, before spring break. So I understand, if you guys forgot some of that, but microbicidal, just like homicidal, it's going to be actually killing the microbes, whereas microbistatic, we're putting them in stasis, and they can't grow. So they can't grow, they're not growing, but they're technically still alive, like, waiting in the darkness to come back out and grow again. So that's what the difference is there. And cellular targets, right. So in the microbial control chapter, as well as in the antibiotic chapter, we talked about the targets of the things we were using, right? Whether it's, our cleaning agent or physical means of cleaning, as well as the antibiotics both. So cell wall, cell membrane, nucleic acids, and proteins were the big four, the main four that they both shared. And remember, the antibiotic one had another one tacked onto it for folic acid synthesis pathway. So that's really the only difference that we got, big picture wise, of how these things actually function on, microbes to kill them or whatever. Yeah, so cell wall agents, here we're talking about detergents and alcohols to break down the integrity of the cell wall. For the cell membrane, surfactants. Remember, they're going to have the charged heads and the hydrophobic tails. So just like they might look like that, and then our phospholipids and our bilayer might look like that. So they're all packed up in our lipid bilayer, whether it's our lipid bilayer or a bacterial, membrane. They're built essentially with the same concept going on. So surfactants can kind of squeeze their way in between and eventually cause leakiness of that cell membrane. And that's important to remember too, because whenever we go on to talk about how the immune system, fights things, one of the things that it does with a lot of the stuff that it does, whether it is through the complement pathway causing cytolysis or through the CD8 positive killer T cells using perforins and granzymes, well, both of those are all involving, poking a hole in the membrane, and stuff can leak out, and that's how we can kill or at least cause enough damage to these organisms for these agents to be effective. So that's one way we can look at that, right? Moving on to the nucleic acid synthesis and function. These will cause damage and denaturation, anything that causes damage or denaturation to the actual nucleic acid. What ones actually do this, though? That's going to be like the radiation, right? Radiation, those rays are going to be literally bombarding the DNA and causing damage. In the case of ionizing, we're like almost removing actual electrons and atoms and causing severe damage to those molecules. Whereas in UV radiation, we're going to be just causing those thymine dimers, right? The molecules to bind next to each other instead of across from each other like they're supposed to. And then we can't read the DNA and replicate it correctly. So that's the nucleic acid ones. In this portion, in the microbial control, we didn't really talk much beyond that about things, affecting nucleic acid, but we did a little bit more in the antibiotic chapter. All right, and then the proteins, anything that causes denaturation of the proteins. And we focused the most on heat, right? Whether we were talking about that moist heat, which you need a lower temperature as long as moisture is there. But if you don't have moisture, or you can't, because some things can't have that repeatedly, like some types of metal, because it'll rust and damage over time. That you can still use dry heat, but you're going to have to do that at a much higher temperature. And the way that would work to basically get rid of the microbes, to denature those proteins, often has the effect of incineration. So actually burning those microbes away. That is denaturation of the proteins, breaking those molecules down to where they're no longer recognizable from what they were before. And that's permanent damage that can't be repaired. So just like when we flame air loops in the lab. So that is chapter 11. And if you guys have any questions about anything dealing with chapter 11, let me know. Sorry, I'm looking at my notes too to try to make sure I'm getting what I want to talk about. Right, we did this. Oh yeah, and I do like this little, I'm trying to draw with a pen like I do in lecture. I don't have the pen, I have my mouse. The disinfection, sterilization, antisepsis, and decontamination. So again, I just drew right through that, but antisepsis, that's going to be the body surfaces, that's important to know. Decontamination or sanitization, that's mechanical removal. And then when we look back at disinfection, destruction or removal of vegetative pathogens, but not bacterial endospores. Usually we're referred to only on inanimate objects. So this is not just talking about mechanical here. So that's the difference between disinfection and decontamination. Decontamination, only the mechanical methods of removing things. And then disinfection, talking about any way that we be removing it, chemical, physical, whatever it is. So I did want to talk about that. So, all right, chapter 12, we talked about drugs used to control microbes. So remember, antimicrobial chemotherapy. Chemotherapy refers to anytime you're using a drug to treat a disease or even symptoms of a disease. So in this case, antimicrobial chemotherapy, we're talking about antibiotics and similar drugs like that, right? Antimicrobe, right? So I brought up antibiotics because it's the one that this chapter really focused the most on, but it could be, anti-protozoal, it could be antifungal, it could be any of those. Everything falls under antimicrobial, but so the test that we did in lab that I keep freaking talking about, but it is important to remember that this is an important test for understanding the susceptibility of a microbe to a chemical chemotherapeutic drug. Using that Kirby-Bauer assay. So, plating something out on a plate, nice and smooth, like a big lawn of bacteria, before you incubate, popping on those discs of whatever drug you're interested in, and then, incubating it to grow, and then seeing if we have zones of inhibition, right? Zones of inhibition being those clear areas around the disc where we didn't see growth. And those just mean that that's where the microbe couldn't grow. You have to do more testing to see if that drug was microbicidal versus microbistatic for that drug on that microbe, right? So we remember all that, taking the sample in the clear area and restreaking it again. So remember those concepts for that. Therapeutic index is a term referring to the ratio of the toxic dose of a drug to the effective dose. So how much you need to take in order to get rid of the microbe is the effective dose. How much you would be taking in order to see negative side effects starting to come out. That's the toxic dose. and you want to keep those as far away as possible. The closer they get to one another, the more dangerous it is. Okay. All right. Selective toxicity does not have anything to do with the therapeutic index. I just want to be clear about that. This is something that's not related to that. Selective toxicity is referring to like when you're making a drug or designing a drug, you want, well, I guess it is related a little bit, but I don't want you guys to confuse too much with those concepts. So when you're designing a drug, you want it to kill microbes as much as possible. And you want them killed ideally, because then they can't come back later or develop, any resistance, anything like that. So that's why we would want them killed as opposed to microbicidal, right? So we want it microbicidal with the least amount of side effects on the person that's taking it. So yeah, it is related to the concept of therapeutic index, but it's more or less talking about toxicity for both the microbe and the human at the same time, taking that into consideration. So that's selective toxicity, and that's an extremely important concept in Chapter 12. When you're considering, administering any antibiotic to, any of your patients for any reason, you want to weigh that concept. How sick your patient is and whether or not there are side effects of that antibiotic or other, chemotherapeutic drug, whether they could handle it. That's giving that selective toxicity, that therapeutic index, those things a little bit of thought. So moving on to the drugs themselves, antibiotics can target cell wall, cell membrane, nucleic acid, and protein. So I put them in the same order as the other one, just that you can see that it's the same. And then we also tacked on folic acid synthesis as another target for antibiotics. Okay, those are our main targets for these. I'd be familiar with these main groups that I've listed here in the review. I know there's a lot of drugs on here. I'd be familiar with these main ones, FYI. So cell walls, things that are going to be targeting cell walls, typically synthesis of the cell wall. The silins, those like penicillin, amoxicillin, ampicillin, anything ends in silin, they have a beta-lactam ring. That's a square-shaped one that we saw. And that structure of that molecule is important for its function. Whenever some bacteria are resistant to these antibiotics, they actually break down that beta-lactam ring, and that's what makes it not able to function anymore. So important. And then we have, so those are the beta-lactams. Then we have cycloserine, cephalosporin, vancomycin, bacitracin, isoniazid. I would know that they are the cell wall medications. Definitely know that penicillins are cell wall medications and that they're associated with beta-lactam rings, okay? Okay, so that's the cell wall stuff. The cell membrane, the only ones I need you to be aware of, and I don't need you to worry too much about how these function. Just know that they are associated with affecting the cell membrane or the cell wall or the proteins, whatever it is. Know that they are affecting those things. So if I were to give you a question, what aspect of the bacteria does polymyxin, treat or damage or whatever, and then you should be able to tell me it's the cell membrane. So recognizing it that way at that level, I don't need you to tell me how exactly these work or any of that. And these are the ones that I feel like were the most important to be aware of. Definitely the ones in bold are the most important. So if you need to focus on anything, focus on the ones in bold. So cell membrane, ones that's polymyxins and daptomycin. There you go. Nucleic acids, things that target nucleic acids, fluoroquinolones. These usually end in floxacin, like ciprofloxacin, cipro. These often, not always, but often are nucleic acid like analogs. So they look similar to the A, T, C, or G and replace them. And then whenever they do that, it makes it so that the DNA can't be replicated properly. Like if we'd have had a thymine dimer from the UV radiation or something, right? So the body doesn't really recognize it properly when it tries to match it up, and we're making copies. So that's kind of how that works. Those are the nucleic acid ones. The fluoroquinolones ending in floxacin. Proteins. These often affect the function of the ribosome. I do not need you to know exactly how they affect the ribosome. Know that they do affect the ribosome, and that's how they're going to affect the protein synthesis, okay? Aminoglycosides, a lot of these end in mycin, not all of them, but a lot of them do. And then tetracyclines and glycylcyclines. So those are pretty big ones to be aware of as far as the ribosome affecting medications go. And then we have the folic acid ones. That's things like sulfas. So sulfamethoxazole, that is trimethoprim. So be aware that trimethoprim is a sulfa drug. I would definitely, remind yourself of that. That's just a name of the drug. The chemical aspect of it involves, sulfur in it. So those sulfa drugs, any of them, sulfa whatevers, and trimethoprim, which is also sulfa something, they all are going to step in that folic acid pathway and block it somehow, usually as a competitive inhibitor, which means that they're going to bind at that substrate site so that the actual substrate won't be able to do that. And now the enzyme can't act the way that it's supposed to. So that's how that's going to go. All righty. Oh, geez. Any questions about that part before we move on to like everything besides antibiotics? Everything besides antibiotics, we'll go through those. That's the antifungals, the antiprotozoals, treating helminths, all of that stuff. The antifungals, these are going to include macrolides and polyenes. That's just the name of the drug. And I don't need you to know too much about the structure of these or whatever. But things that are like azole at the end, like ketoconazole, a lot of those are going to be related to antifungals. And we do see that, that term at the end of some other drugs that we use for other things, but they have the same chemical concept. So that's why their name is the same thing. But a lot of these antifungals will have azole, like ketoconazole. If you've heard of amphotericin to treat a fungal infection, that is a macrolide or polyene. And then we have the echinocandins, which often end in blah, blah, blah fungin. I don't know much about these, but there it is. So the antiprotozoal and antihelminthic drugs, I would be aware of these too. Quinine, anything ending in quin. I don't remember like the names of the medications off the top of my head, but it's bloquin, like that, Quine. These are drugs that are going to be treating malaria. All right, so quinine based drugs will treat malaria. And that's a protozoan, just to be clear. And then we have metronidazole as well. That's a pretty widely prescribed medication for a lot of different kinds of infections. Like any anaerobic bacteria can be treated with that as well, but it is used to treat protozoan, eukaryotic types of infections. So there's that. So that's really all you need to be aware of for those. The antivirals. These medications are often very virus specific, and I'm sure that you guys learned that whenever I was going over this in the lecture. Tamiflu and Relenza, both of these are going to inhibit the fusion, entry, uncoding, everything like that for flu. And that's it. That's it, just the flu. So those will only affect the flu because the flu has this molecule on its surface. When we talk about H1N1 or H5N1, I don't know all the numbers or whatever, but those the N portion is neuraminidase, and so you can have all these different versions, N1, N2, N3. So the neuraminidase is a molecule on the surface of the flu virus that whenever the flu virus is budding out of a cell, will help clip it free. It also, so that's one important thing that it does. The other thing it does is whenever it is getting into the cell, it helps with like attaching to that same receptor and allowing it to be taken in and, unsheathed and allowing all the DNA and everything to get into the cell to replicate. Okay. So that is the idea with neuraminidase. and Tamiflu and Relenza block the function of neuraminidase. That's what they do. And so that is why we can only use that for flu, because other viruses don't have neuraminidase. That's unique to flu. That's the same thing also with a lot of those HIV medications. A lot of them target reverse transcriptase. There are other viruses out there in the world that have reverse transcriptase, but they're not like pathogens, and especially not the way that HIV is. So all of those drugs that target that aspect of it, they're only going to treat HIV. They're not going to help any other aspect of anything. And if you go through all of these drugs, you would see that the way that they function have a lot to do with the specific structure of the virus and or how they associate with some sort of receptor or other protein in the cell. So very, very specific. And that's why those antivirals are specific to specific viruses. Okay, so that's the concept behind that. So we talked about the ones that inhibit entry, getting into the cell, basically getting in and starting everything, entry, fusion, uncoating. Those are the first kind of steps of infection, right? The ones that inhibit transcription or translation, we talked about acyclovir and other cyclovirs, which is used to treat herpes infections, or ribavirin as well as used for that. So reverse transcriptase. Reverse transcriptase is another target. So that tends to be when we are targeting things like reverse transcriptase in HIV, nucleoside or nucleotide analogs. Either one of those building blocks, those A, T, C's, and G's, being similar to those, they're incorporating those incorrect ones that's associated with the drug, the antiviral, so that the actual real ones can't do their job anymore correctly. So that's how that would work with those nucleotide analogs. And then we have the ones that prevent maturation and budding. This can also include like Tamiflu, because it also has action here, as well as inhibiting getting in. But the ones that we also talked about were Endinavir and Saquinavir, I guess. I don't know the names of these, but I can tell you about that Saquinavir, whatever it is, how it works. So HIV, in addition to having its special little reverse transcriptase, which helps it put its turn its RNA into DNA so that it can put that DNA into our actual genome. In addition to having that, they have something else called protease. And what the protease does, basically, HIV proteins, for everything they need to make for their little viral particle, they make it all kind of in a long strip of like proteins, like kept going, we never got the stop codon. So we make it all in one big link, and then we clip it apart into what we need. And that's how HIV has that made. Other viruses can do some similar things, but for HIV, its particular protein that does this is, we call it the HIV protease. So we have another thing that we can target specific to HIV infection that doesn't involve our proteins, and that's what I guess Saquinavir does, but that's how that works. Okay, that's the antivirals. So the big thing to remember there, the ones that inhibit entry, the ones that inhibit transcription or translation, DNA stuff or RNA stuff, and the ones that prevent maturation or budding, so building up the virus or getting out of the cell as a virus. Okay, drug resistance. We did talk a little bit about that. That can be intrinsic or acquired. Intrinsic is just talking about if you are a bacterium that produces enzymes that break down, penicillin, right? You might have those enzymes if you are somebody that makes penicillin just naturally, because you don't want to get affected by it, right? So, having a defense mechanism against the drug that you make as part of your natural life cycle, that's intrinsic resistance. Acquired resistance, that's what we would talk about getting from horizontal gene transfer, whether we can get that through transformation or conjugation or transduction. I know you guys are sick of hearing about those, but conjugation is whenever bacteria are joined physically to one another through the sex pilus and can transfer things physically across that little bridge that they made. Transformation is whenever bacteria, competent is the word for them, bacterial cells can take DNA from outside of themselves. They could be whole plasmids or even bits of DNA and take them up into their cells and either incorporate them into the genome or just express them due to them being a plasmid and having their own, operating system essentially. And then transduction, which is getting genes because of getting infected with viruses. Phages, right? Bacteriophages. Bacteriophages, of course, being viruses that infect bacteria, for those of you who can't seem to hold on to that concept. I totally lost where I was for a second. So that's basically, acquired mutations can also be, so that's horizontal gene transfer that I talked about. You can also get mutations through spontaneous mutation, of course, right? So how would a bacteria even get those mutated genes to allow them to transfer that to other bacteria? Well, it had at some point to be a random mutation, and now it's getting transferred that way elsewise. So spontaneous mutation, again, just happening due to random, mistakes in the genetic code. Some of them happen to be beneficial. Those ones that are beneficial, those bacteria are more likely to survive and pass those on to, their, I won't say offspring, but whenever they duplicate, the resulting bacteria from that. So yeah, we get that. So probiotics and prebiotics and fecal transplants. Of course, probiotics are those bacteria that will be healthy and good for our gut to function the way that it should, nice and good and happy and easy. Prebiotics are like the food for them. And then fecal transplants are something we could do for someone that needs a new gut biome, usually due to antibiotic treatment to get rid of microbial infection in the gut or microbial infections anywhere else. You take antibiotics, it clears out all the microbes in the gut. Maybe they're left with nothing, roughly, or maybe they're left with something like C. diff or something like that. And we want to, give them a better chance to not get C. diff infection. Which is called a super infection. Yeah, so we've talked about that a lot too. So we give them poop from somebody else, usually under anesthesia, through colonoscopy in order to replace that biome for them. Okay, drug toxicity, that's just side effects from the drugs. We did just be aware of the fact that there are side effects. I don't need you to know every single friggin' side effect of every drug that there was in that chapter. Be aware that some drugs do have toxic side effects, right? Some of them can be pretty severe. You can develop allergies to these drugs, like what we see with sulfa drugs and penicillin. And the super infection aspect, you treat somebody with an antibiotic, you clear out their natural biome, and now we have overgrowth. Like we said with C. diff, like you can have when you get a yeast infection. Take an antibiotic, clear out the normal flora of bacteria, the yeast will overgrow. Yeast are normal to have in that area. In women, so that's not a weird thing to have down there or anything, but it'll overgrow if you get rid of the bacteria that are like, taking up the stadium seats that now the yeast is going to take up whenever you get rid of the bacteria. So that's the idea with that. Okay, any questions about chapter 12? I don't know if you guys are, yes.
Sorry.
Yeah, you're fine.
Question. Would you say that those antivirals, the inhibiting and preventing ones, are examples of prophylactic drug use?
You can use them prophylactically. So that is a good question. You can use them prophylactically, especially with herpes. So if you were, getting in a relationship with somebody who has herpes and you know that you do not have herpes, they can prescribe you, anti-herpes, prophylactic. And basically it's the same medication that they use to treat herpes, but they just keep that in your system so that the virus is less likely to be able to, grow and take a foothold in your body to protect you from getting that disease. They do that with HIV as well. If you were in a relationship with somebody who had HIV, they can treat you with medications for HIV so that the virus can't get a foothold in your body. And they work pretty well, actually. So good question. All right. Familiar with C. diff. I would say C. diff and yeast. Those are the two big ones. My internet connection is unstable. I don't understand how I'm sitting right next to the frickin'. You're okay. Yeah, I would say yeast and C. diff, but yeah. Those are I feel like they're kind of a little bit obvious ones too, so not too tricky there on me. Okay. All right. Infection and disease, right? So we talked about like basically what it is to get infected and how disease progresses and everything like that in chapter 13. Important to understand infection terms like infection, pathology, disease, infectious disease. Disease can be anything where the body is deviating from the norm. That includes, having cancer or diabetes or something. So then in this case, in our, microbiology class, we're mostly talking about infectious diseases. So it's a different thing. So be aware of the difference between just disease and infectious disease. Right. So infection is anytime you're going to have like introduction of growth of the bacteria into an area, typically where they shouldn't be growing. Sometimes you have symptoms from that and sometimes you don't, but anytime that they are growing there, that's an infection. All right, pathology would be like development of those diseases, just the symptoms, essentially. Okay, resident biota and microbial antagonism. This is that concept that I was just talking about with like taking up the stadium seats. Your resident biota are essentially bacteria that we would call good bacteria that are doing a lot of things for your body, whether it is helping you break down things in your body or protecting you from, other bacteria that could be coming and invading, and all that stuff, strengthening your immune system as you develop. Everything's great about the resident biota, especially the gut biota. That idea of microbial antagonism, that is to say that your normal biota, especially like your gut biota, are taking up those seats so that bad guys can't come in and overgrow. So I can tell you all day long that if you have C. diff normally in your colon or yeast normally, in your genital area, that whenever you take an antibiotic and we get rid of all of those, normal bacteria that would be living there, now those super infections can occur, what we talked about in the previous chapter, but why don't they happen when we're not taking an antibiotic? And this is why it's that idea of the microbial antagonism. The normal biota that is there is taking up the stadium seats so that the bad guys can't sit there, as well as they're also sometimes producing toxins to keep the bad guys from being able to grow and other things like that. So that's the important aspect of the resident biota and kind of what it does for you in protecting you from infection. How do you get your biota? We talked about essentially as a baby, you might have some biota associated with being a fetus in the womb. And then whenever you're born, you're getting exposed to the, bacteria of the vagina, or you're getting exposed to bacteria of the skin associated with a C-section, or, any other thing you would be touching first in your life that might not be sterile. Those things will grow on you and can become part of your normal biome. And then breast milk can do the same, and you can even introduce similar bacteria into your baby through probiotic type breast milk to help with that function. So, you get that. Okay, so that's where we get that from. Let's go through the infection stuff. We have our portal of entry. A portal of entry could be exogenous or endogenous. Exogenous portal of entry means that something came from outside. That's obvious. Somebody sneezes, you get sick from it. All right, that came from outside your body. Endogenous, that's like that C. diff superinfection. Okay, you already had that in your body and it's being overgrown. Or the other example that I gave in lecture for an endogenous infection is getting E. coli into like the bladder and the urethra, where it normally doesn't live there, normally is in your colon, but sexual activity can often spread that into that other area. That's the most common way to do it. Wiping improperly is another way that you could do it as well. But yes, then you would get growth of E. coli in a place where it shouldn't be. So that's an endogenous infection as well. So the next step is, so got in, the next step is attaching or adhering. And typically it's just binding with some sort of molecule surface marker on the host cell. Specific for every sort of microbe, or even could be involved in having like a capsule that lets you be sticky to stick onto things. So direct binding with markers on the surface of the cell, could be general markers that recognize PAMPs, right, that we talked about with some of those innate immune responses, the PAMPs being pathogen-associated molecular patterns, like lipopolysaccharide and stuff like that, peptidoglycan, all those things that your body can see automatically as foreign. Those guys can interact with other cells through generic interactions of that situation. So what else do I want to say about that? So it can be specific or it can be general interactions. So sort of attaching, adhesion. So now we're going to be taken up into the cell, or we're going to just be stuck out on the cell multiplying, depending on what kind of microbe you are. So we have host defenses that are going to come and attack you. We learned about that with the innate immunity, as well as eventually your specific acquired immunity. So now stuff is going to come and start trying to kill you. How would you defend against that? The thing that we talked about, there are a lot of ways that bacteria can defend against that, whether it's molecular mimicry, making themselves look like us, to try to just like, they don't see us, hide, essentially, blending in with the crowd. And then we have anti-phagocytic factors, things that make it hard, literally, for phagocytes to eat up those cells, so that whenever they come to investigate this foreign matter, they aren't able to engulf it and go be antigen-presenting cells, things like that. So that includes capsules and other stuff. So some molecules that the bacteria can produce can help with that too. So next thing, causing the disease itself. This is going to have to do with anything we would call a virulence factor, anything that helps that microbe cause disease in your body and establish disease in somebody. Even if it's somebody, like if we were only talking about like, well, in old people, they are the only ones that get symptoms, even that counts, okay? Anything that helps them cause disease is a virulence factor, anything. That was a very confusing thing for a lot of people on developing their PowerPoint slides, trying to figure out what even a virulence factor is. Well, literally anything, man. A capsule counts as a virulence factor that helps it, establish itself to help cause disease. All sorts of stuff like that. Whether it makes a toxin, that's a virulence factor. Anything. So if it can hide from the immune system, it's a virulence factor. So think about that. So virulence factors. We can also associate one type of virulence factor that we can have is a toxin, right? There are exo and endotoxins. Exotoxins are produced by the microbe, usually bacteria. Like it's a protein, usually an enzyme that they secrete outside of their cell to fight off, the immune system and cause damage to tissue, to allow better invasion and everything like that. Those are exotoxins. Okay? They usually cause some sort of damage to our cells. Endotoxin is associated only with gram-negative bacteria. I'm sure you are all saying it with me out there. Probably not, but that's okay. I forgive you. But yeah, so gram-negative bacteria, they are the ones that have endotoxin. So it's part of their membrane, outer membrane. I want to be clear about that. The outer membrane, part of the lipopolysaccharide molecule. It's lipopolysaccharide as a whole, but part of that is the endotoxin molecule. So that can, I won't say break off, but be recognized as the endotoxin molecule whenever the cell is being destroyed or just died on normal reasons, whatever else, broken apart for any reason. Okay, so signs versus symptoms. Signs are the ones that are measurable, like a fever. You can actually palpate somebody's swollen lymph nodes, so that counts as a sign, versus symptoms that are being reported by the patient, but you can't necessarily verify it yourself. So things like pain or feeling tired and stuff like that. And then we talk about syndromes. Syndromes are things like meningitis was the example that I gave, and I think it's a pretty good one. You can have meningitis be caused by all sorts of like bacteria, viruses, and fungi, but they all have essentially meningitis symptoms, and that's why they're meningitis. So meningitis is a syndrome. It can be caused by a lot of different things, though. So that's an example. Cold, right? The cold is a syndrome. what can it be caused by? There's like 900 viruses or something that could cause the freaking cold. So you never know. You never know which one you have. Okay, so we're starting to get sick. And here are our signs in terms of sorry, I keep wanting to say syndrome. Signs and symptoms. Why would they be happening? What's causing those to occur? Okay, so we have development of inflammation, which we did talk about in the next chapters a little bit more. Leukocytosis or leukopenia, leuko being white blood cells, all of the white blood cells. Okay, it can be any of those white blood cells, includes the lymphocytes. Okay, septicemia versus bacteremia. Septicemia is growth of the bacteria in the blood. Flourishing is what it says, but flourishing of the bacteria in the blood. Bacteremia, that's any, like if you just had any bacteria in the blood. And then viremia is essentially the same thing for viruses. Okay. Asymptomatic and subclinical infections can occur. We know this. This isn't uncommon for some infections, right? You can have be infected with something but not show any symptoms. That's all this is talking about. For whatever reason it is, their body is just reacting differently with their normal immune system, or you're like a healthy person, so you don't have symptoms with this thing, but people who have a bad immune system, immunocompromised people, older people, pregnant women, children, and people with actual immune diseases would be susceptible to the symptoms or whatever. So that counts too. Latency, that's like with the chickenpox or with herpes, like herpes infection. You have your outbreak with the skin lesions, and then it goes away for a while, and then maybe you're stressed out or your immune system drops just enough that it can flare up again. That's an idea of an example of latency that can happen. And then sequelae are basically anything that lasts past the infection, so effects of that infection lasting even though you've cleared the virus, anything like that. So so COVID does a lot of that, right? Heard a lot of that from COVID. People clearing out the virus, but still having effects. Those are sequelae. All right, how do we get out of the host? So we've grown, we have fought off all of the host cells, and we have caused disease with all of these signs and symptoms. And now we're going to exit the host. And how do we do that, right? I don't know, you guys know how. It's like through the mouth by sneezing or coughing, or through the nose by sneezing, or I don't know, poop or urine or blood. Any of those things can lead to exiting the host. You know how things get out of the body. And then the 4 phases of infectious disease, that's going to be incubation period, right? So the incubation period where the organism, the microbe is growing, but we're not actually seeing disease, right? Symptoms or signs yet. So that's the incubation period. It's just starting to grow. It's just starting to get a foothold, but we haven't seen the immune system respond to it yet, which is often where our symptoms come from. Next, we have the prodromal phase. That tends to be the first initial signs and symptoms coming up. Now we're starting to just get inflammation going or fever developing and whatever. So that's the prodromal phase. Invasion is where we're full on with like the worst of the symptoms that you can have in that infection. And then convalescence, we're starting to clear the virus, and therefore the symptoms themselves are waning. Okay, we talked about reservoirs and sources and carriers. And we talked about zoonosis. I don't really like this chapter. It's just a whole bunch of stuff all together, I feel like. But reservoirs, just places where the microbe just exists naturally, and it may not necessarily be causing disease, like in an animal population. Like if you were to talk about, certain viruses just living in bats, and bats are fine, but they eat a piece of fruit and it gets dropped on the ground, and then a pig eats that, and then we eat the pig and we get sick. That's, bats would be the reservoir. They're not getting affected by it. Okay, we have the source. Source is just what it sounds like, where the infection would have come from. The carriers, those are going to be usually people who are not having many symptoms or subclinical for whatever reason, and can be transmitting the disease. So that can even include people in that convalescent phase. You think you're feeling better from COVID and you go back out and you're still contagious. So that's kind of what this is referring to. Anybody that is going to be able to carry that virus around and likely to spread it around. And then the zoonosis, anything that can be from animal to person. And a lot of times like rabies. Of course, that's my favorite example, but rabies, typically exists in the animal population. And if a person gets it, then that's a zoonotic spread. Transmission, communicable, contagious, and non-communicable. Okay. Communicable means that person to person, it can be spread, right? It can be transmitted from one person to another person and maintained potentially in the population. Contagious means that it is very much able to do so. And then non-communicable just means kind of what it sounds like. You're not going to be able to spread that disease from person to person. Like whenever you scrape your knee and it gets infected, like you're not spreading that infection to people. So that's non-communicable. Pretty basic example, but it works. All right, we can spread things through vehicles. The main example that I want you to be aware of is fomite, right? If I have, my phone here and, I am talking on, oh, Jesus, sorry, I just forgot I had phones. I'm talking on my phone to somebody and I'm touching it and I have COVID and I'm coughing and oh yeah, okay, thanks, mommy. I'd love that chicken noodle soup. It sounds great. And I put the phone down and then, my husband comes along and grabs it to call somebody or to look something up on my phone. And now it's been contaminated with COVID. Well, he probably would have already had it, but you get the idea. So that's a fomite. The thing itself is not, got any disease associated with it, but we're putting microbes onto it, and then that is then getting transferred to other people who are touching or interacting with it. So that's the fomite. Then, of course, oral fecal spread, not washing your hands enough when you're going to the bathroom, and then touching other people's food or your own food even. Water and soil spread. A lot of stuff like Giardia is in water. Tetanus in the soil. Like there's just a lot of stuff that can just naturally be there. And then the air and droplet, like the air transmission, whether we're talking about droplet nuclei and aerosols, those being like kind of drying up particles from sneezing or coughing, but they're going to be a lot more long-lived than the normal direct transmission of coughing and sneezing and those droplets. So, but yeah. So those are a lot more long-lived. And then we have nosocomial infections that we talked about. That's just any sort of infection that you can get or are more likely to get in a healthcare environment. And then epidemiology terms that we went through. Reportable diseases, anything you can or must report to like the CDC or your local government so they can track that disease and make sure they are keeping an eye on it. Prevalence and incidence, basically a big picture idea, prevalence, whereas incidence is dealing with like a specific situation, like breaking it down to like this month, how many cases we had or something like that. The morbidity is referring to, how many people are sick, essentially, and mortality, how many people died from it. Point source versus common source. Point source is referring to a single, like even person that the disease can have come from. We know it's one specific thing, whereas a common source, it can be things that are kind of spread out over a larger area, but are still associated as the same thing, like water wells were back before they figured out about controlling water contamination for cholera. But there might be several water wells that would be affected in a town or city. But yeah. Sorry, I have a little thing I have to drag out of the way. Epidemic versus pandemic, right? So epidemic would be like a location, basically, whereas pandemic is a whole world. So then endemic versus sporadic. Endemic is something that is natural in that area, and sporadic is going to be like it randomly pops up in the area. So when we had Ebola coming over into the United States, that was a sporadic outbreak. Then we talked about emerging and re-emerging diseases. Of course, emerging being things that are still new to us relatively as a human population, and re-emerging being those that we saw basically a decline in, and now due to change in society or something, we're seeing a re-emergence of it, whether that's due to global warming, which is often contributing to increase in mosquito-borne, vector-borne diseases, and then moving a little bit further north. Or it could be, like tuberculosis, where, we're getting spread of tuberculosis among the HIV population, and then they could be spreading it to other parts of the population. That's an actual thing. Okay, this is a lot of words, and I have this thing in the way. All right, non-specific immunity, our innate immunity. So our first line of defense stuff, that's our physical and chemical barriers. The skin, your tears, all of that stuff that's on the outside to keep things from getting in. Mucus, any of that. Lysozyme is an enzyme. Remember, enzymes are proteins that break down other molecules. Lysozyme is an enzyme that breaks down peptidoglycan. So it'll help attack and break down bacteria that are invading. So that's sort of like our first line of defense. We can see lysozyme in like tears and saliva and stuff. So the immune system, let's go into that, the second and third line of defense, right? This is mostly this chapter dealing with the second, the innate part. So either way, we have to be able to recognize self versus non-self. That's done by looking at surface molecules of, our cells. So it's just surface markers that allow us to know whether it's self or non-self. There's a lot of them, but there you go. That's really all you need to know. Surface markers. Lymph system, the lymphatic system dealing with the thymus, the lymph nodes, the spleen, and the lymphatic fluid, just those four parts is what you need to be aware of, as well as the cells themselves, of course, that are associated with it. All right, then we talk about the white blood cells for our immune, knowledge. We have, these are all called altogether leukocytes. Then we break that down to the granulocytes and the agranulocytes. So the granulocytes include anything fill, neutrophil, eosinophil, basophil. So that's those, right? We also have the phagocytes. So a lot of these, neutrophils are also considered phagocytes. And then other phagocytes are going to be our monocytes and our macrophages. The mast cells are included, those nasty mast cells involved in your allergies. And then the dendritic cells. So those some of those become those antigen presenting cells. Then a lot of these cells are going to act by recognizing those PAMPs, those pathogen associated molecular patterns, anything that seems like it's something that is associated with foreign. So that's going to be things like lipopolysaccharide or peptidoglycan, and all sorts of stuff like that, that our cells just automatically recognize as if they see that molecule, then this must be foreign. Let's take it in and start breaking it down, and then we'll start presenting it as antigen presenting cells to see if our T cells can get activated by it. So that's the idea of what's going on there. Phagocytosis, the steps of it. We have chemotaxis. What does that mean? We had a signal somewhere in an area by cells maybe that were damaged, or other cells like other phagocytes or something in the area can also release those chemokines or cytokines, basically those call to arms chemicals. We sense those as cells, and now we're moving in towards that. That's chemotaxis, right? We heard the call. And then adhesion. So we're going to stick to an area literally, and that's going to be adhesion of, stick to an area of the microbe. So it's typically going to be some marker on the outside of the microbe, usually the PAMP for these cells, right? So in order to phagocytose them, we're going to adhere to them somehow, or they will adhere to us. Typically, they think they're going to infect us. Good luck. So that's what's really going on. But we're going to engulf them into a little thing called a phagosome. The phagosome is just like a vesicle inside of the cell with membrane around it and your bacteria inside. Then that's going to mix with a lysosome. A lysosome carries lysozyme, go figure, and other enzymes as well that are going to be used to kill that microorganism. They fuse together and destroy and kill that microbe, and then we will release any of that debris that's left over. Sometimes we can use the stuff from those cells though, so whatever, but that's phagocytosis and the steps involved. Agranulocytes, those are the ones that in, you know, a microscope aren't going to be having, you know, what they look like granules. So these includes our monocytes. And then once they migrate, the cells in the bloodstream, when they migrate to a tissue, that's a macrophage. It develops into a macrophage. If it goes into the skin, it will further develop into a dendritic cell. So they're kind of all related with one another, okay? Yeah, so those are the big three there. Those are all antigen presenting cells, or APCs. Okay, so those are agranulocytes. Then we have the lymphocytes. Those are also agranulocytes. And remember, lymphocytes are just the T cells and the B cells. All right, we'll talk about those in the next chapter. The inflammatory response. A lot of this is triggered by our innate immunity. That's what they are doing. That's like part of that first line of defense, or second, sorry, I guess it's second, technically. The skin was the first. But part of that initial defense by our immune system is generating inflammation, and for a reason. And the inflammatory response itself involves rubor, which is redness of the area, or, you know, whatever. I guess if it's on the surface of the skin, you would see that. But redness, calor is warmth, tumor is swelling, and dolor is pain. All of these things are associated with the inflammatory response and are happening for a reason as a result of that inflammatory response. They are sort of like side effects a lot of the time. That we're not necessarily trying to start, you know, damage or pain in the body, but it's a side effect of us trying to fight off fighting off of a microorganism or invader of some kind. So a fever is part of all that, right? So fever, these are triggered by molecules called pyrogens. Endotoxin, for example, is a pyrogen. There's a lot of other ones too, but endotoxin is a good example. This increases cell metabolism and activity. It decreases microbial growth, and it causes the release of transferrin, and microbes need that in order to, you know, survive. So keeping those things away from them. So it's three things that fevers can do for us that are positive. These fevers can also be caused by exogenous sources, like an infection, you know, like peptidoglycan or endotoxin was the best. I guess I should have just said that, but that's a great example of an exogenous source. That's not part of our body necessarily. So that's exogenous, whereas endogenous signaling, that's signals that are transmitted by our cells. Those call to arms signals, that's what we would consider endogenous signaling to cause fever. Okay, we can sometimes send those signals out when the cells see something that they think should warrant them triggering a fever, but it itself isn't doing that triggering. The cells are. Okay. Moving on to interferon. Remember, interferon was like the one molecule that I wanted you guys to be aware of those like cytokines and chemokines. Interferon, in this case, now that's our defense against viral infections. That was whenever, you know, Larry called Tom to tell him there was a burglar in his house and to get the shotgun out. That is telling the cells nearby, there's damage going on inside of me. You should probably start making some proteins to fight against that. And then that's what happens. So that's interferon, interfering with the, you know, burglar's activity. So only for viruses. Then we talked about the complement cascade, which I think is just one of the coolest thing. It's so useful even in non-specific immunity, but of course useful whenever we're talking about you know, antibody mediated sorts of responses, because the first type of complement cascade that we learned about was the classical version, right? And that was triggered by antibody. But anyways, in the complement cascade, the steps are initiation, amplification, polymerization, membrane attack. That's just basically how those proteins in the whole cascade are going to be broken apart and work together in order to eventually build those holes, poke those holes into the membranes for cytolysis, right? Cyto being cell and lysis being breaking apart. So we had the three ways that you can trigger complement cascade. The classical way being triggered by the presence of antibody. The alternative way being triggered by the presence of LPS, lipopolysaccharide, associated with bacteria. And then lectin pathway. And lectin is a protein that can bind onto mannose-containing molecules on the surface of certain bacteria. So another thing that's not, it's pretty similar to like LPS, but we're talking about a sugar now. But lectin binds with that. So those are the three ways you can activate complement cascade. And then what the complement does for us is it can lead to opsonization. It can lead to opsonization. So helping the phagocytes eat up whatever it is, the microbe, right? Maybe it had a hard time eating before. Now we can activate our complement cascade, and those proteins that are involved in that can help the phagocytes eat up and then maybe go to antigen presentation or just killing things in general. That's opsonization, helping out phagocytosis. Cytolysis, we just said about that, poking little holes, putting those complement proteins together as holes in the membranes of the target organism, and causing stuff to leak out and leading to cell death. And then inflammation, which we talked just previously about the importance of inflammation. It's also often a call to arms reaction. The more cells we can get in there reacting, the more macrophages or dendritic cells that we call to an area, the more likely we're going to pick up the right antigen to signal the start of our specific immunity and all that stuff. So that's kind of the idea with inflammation as well. Let's move on to chapter 15. Does anybody have any questions about 14? Oh God, I forgot this. I drew this. I thought I put this after 15. Apparently I did not. I'll go over that if I need to in a minute. It's really bad handwriting. I forgot that. Okay. This is a lot, this chapter, right? This was the big daddy chapter for this unit. Chapter 15, dealing with specific immunity. Introducing a lot of cells and concepts to you guys that you knew nothing about previously, but that I hope that as you guys learned about them, you thought it was at least a little bit interesting, and saw the usefulness in it, I hope. But okay, so lymphocyte development, everything starts in the bone marrow. Everything only starts in the bone marrow. B cells will stay in the bone marrow as they are developing. T cells move into the thymus to develop. And then after they've done that, they can circulate in circulation and congregate in lymph tissues as well. So both of them, that's pretty much both of them at once. Okay, we've covered that. Lymphocytes, they are always activated. Whenever they are activated by whatever cell they're being activated by, they first proliferate, which just means multiplying. And then those multiplied up cells. Did you see that? You tell me that you saw that. Jeez. It's the wind, I think. It's so windy outside. Sorry, I just lost my train of thought because of that. They differentiate after they've multiplied. So it's multiplied up cells. We've got tons of the same cell now. Now they differentiate into memory cells, effector cells, or regulatory cells. So that's what's going to be going on there in those lymphocytes. Whether they're B or T, it doesn't matter. Okay. The B cell receptor versus the T cell receptor. The B cell receptor is pretty much an antibody stuck on the surface of the B cell. I don't know what else to say. It also has on its surface MHC class 2. Well, it can express on its surface MHC class 2 when it is presenting antigens to as an antigen presenting cell, because it can do that to the T cells, usually the T helper cell, to activate them. So that's another thing that it can do. The T cell receptor, man, it has similar, variation that's going on in the top, and then it's pretty stable and constant at the bottom. But it will be kind of associating with CD3 is kind of a helper molecule involved in the interaction with the T cell receptor, which interacts with the antigen itself when it's being presented to the T cell, either on a MHC1 or an MHC2 molecule from whatever cell is presenting it, whether it's an antigen presenting cell or even just a normal body cell. All cells have MHC class 1. So, so whatever is being presented and it interacts with that T cell receptor. That's my point. It recognizes that specific antigen. CD3 helps with that interaction. And then CD4 and CD8 will be the ones that will help as well with that interaction, verifying the MHC and all of that. Now, CD4 is present on the T helper cells. CD8 is present on T cytotoxic cells. Okay, that's it. Like they don't like mix and match. So do know that CD4, that's the helper cells. CD8, that's the cytotoxic cells. I don't know why I think a 4 looks like an H to me, so that's how I think of helper, but whatever. It's got that cross. It's probably stupid. Whatever. So that's the T cell receptor and the B cell receptor. So clonal selection, that's referring to removing anti-self clones. We have so many types of B cells that we can have whenever we're born. And then in the bone marrow, as we're developing, we want to get rid of any of the ones that would develop to any self molecules. So as we're developing, that's the hope, is that you're getting rid of as many of those anti-self molecules as you can. And then what you're left with is your typical repertoire. Then we will only be activating the specific one that acts to one specific antigen, not all B cells, right? So that's the important part to know about that, really, more than anything. But clonal selection, remove anti-self and only proliferate the appropriate clone, the one that recognizes that specific antigen. You can have multiple antigens, of course, being reacted to in an infection, of course, but that one B cell, that's all you need. So, okay, antigens, they will provoke immune response and must be seen as foreign somehow. And how do we classify something as being foreign, and how can they meet our requirements for being foreign and interact with as an antigen? So the antigenicity of something. Haptens are too small and they need help. They need a carrier protein to interact with them so that the B cell receptors can recognize them. Otherwise, they're too tiny. Once we've made antibody against the hapten plus carrier protein, the antibody that's made can see just the hapten. So we don't need the carrier protein after that. But as long as we had it to activate, we should be good. Alloantigens are antigens of other people that our system sees as foreign. A lot of times that's MHC1 of the other person. Super antigens, these are things that super activate the immune system. This is often the case when we were talking about toxic shock syndrome. And a super antigen activation going on there. Overreaction of the immune system to a target. And then we have antigen presenting cells. Again, those are macrophages, monocytes, dendritic cells, B cells that are circulating in the blood, in the body, or hanging out in the tissues, recognize something foreign and are bringing it now to the T helper cells to try to activate those associated T helper cells, the ones that are recognizing the same antigen. Okay, if you are infected with something, an endogenous antigen, you have a virus growing inside of your cell, you will present molecules from that foreign thing inside of you on MHC1. Like I said, all cells have MHC1 associated with them. So, so if you're infected by a virus, you should be able to present the virus or parts of the virus on the surface as, presenting on MHC1 to go check out and activate the appropriate response from T cells. What do I say about that? That includes like cancer-related molecules. Okay, so that's an endogenous antigen as well that is foreign because it's not made the way that it's supposed to be made. So we would see it as foreign and present that on MHC1 as well. So anything from inside of the cell, whether it's infection-related or any sort of other abnormality. MHC2, these are only on our antigen-presenting cells, and that's from stuff they picked up while they were going around phagocytosing things, basically. They are presenting exogenous antigen. They picked it up, came from outside. So outside MHC2, inside MHC1. All right, T cells, let's go into the T cells. And natural killer cells, which are very much non-specific, but they act in the same way as the cytotoxic T cells. So there are T cells, antigen plus MHC1. That will activate our CD4 positive TH1 helper cells. So 1-to-1, that's okay. We got that going. That checks. So that'll activate those will then activate our TH1 cells, our CD4 positive TH helper 1 cells will activate our CD8 positive cytotoxic T cells, the TC ones. Okay, so now they'll do their cell thing. And what do? They make granzymes and perforins. The perforins poke holes, or proteins that poke holes in cells. Don't drop the signal. The perforins poke holes in cells, and then the granzymes can get in. So the perforins have two kind of purposes then, right? Poking the holes in the cells, let things get out. That's bad for the cell, as well as letting the granzymes get in and start breaking things down in the cell. That's bad for the cell too. So that's how a CD8 positive cytotoxic T cell will be acting and doing its job as an effector cell. All right, so the antigen plus the MHC2, these are being antigen presented on MHC2 molecules from your antigen presenting cell, whatever it is. It'll interact with CD4 positive TH2 cells. So 2 goes to 2, right? You know it's a T helper cell, so you know that it's CD4 positive. Okay, so these T helper cells, the TH2 ones, will help activate B cells and tell them, yes, we are in fact recognizing the correct thing. I see it, you see it. Okay, I'm activating you. So the B cells will then become proliferate, and some of them will become plasma cells. Plasma cells produce antibody. They secrete antibody, and that's like, they just pump it out like mad. All right, we also have the development of regulatory T cells throughout all of that whole thing. We have regulatory T cells for the CD4 T helper BS, and we also have memory cells for all of this stuff too, okay? Everything has a regulatory and a memory, just think of it that way. Regulatory, memory, effector. Yes, it says that up there, but just in case you forgot. All right, then the B cells, let's move on to those. These are our ones that are going to produce the antibody. Plasma cells are the B cells that produce antibody. The FAB, that is the variable region of the antibody or the immunoglobulin, which is referring to both the antibody and the B cell receptor, which is basically antibody. But that variable region at the top of it is what recognizes the antigen. And then at the bottom, we have our constant region that determines basically what the antibody class is, and therefore what that antibody is kind of going to do and where it can be found and stuff like that. So antigen recognized by the FAB. Antibody, what does it do? Here's the important thing to know about what antibody does. Neutralization. It just covers the thing up with all sorts of antibody, so now it can't interact with anything. It's covered up. So bacteria covered in antibody can't do whatever bacteria do. Opsonization. We're going to help phagocytes do their job, helping phagocytosis, right? They have molecules on them that allow the phagocytes to grab on easier to the antibody than to maybe the cell itself. Okay. Coding. Similar concept to what we were talking about with the neutralizing, but coding is essentially going to be referring to blocking like a receptor interaction between like a virus and I guess that's what I would call neutralization, but coding would be coding it all over, and then neutralization would be the virus interaction. They're the same thing to me, and you're not going to have to know the difference. Let me just say that. So they're the same thing in my mind. They're blocking whatever that organism is, the virus and the bacteria, from doing what they're trying to do. Whether that's a specific interaction that we see in neutralization or a general blocking of anything in the coding, it's blocking it, okay? Complement system, we talked about with the classical complement pathway already, and it can act as an antitoxin, which is not unlike neutralization and coating, right? We're just blocking the activity of the antitoxin with the antibody itself, literally. So yeah, they do a lot of blocking things. Okay, the different kinds of antibody that we have. IgG, that's our big workhorse. That's the one that's going to do the best and brightest job within like the whole bloodstream all over the place, right? The IgM is what we produce first. IgA is associated with mucous membranes. IgE, that's going to be our allergy and parasite associated one. And then IgD is a receptor, the B cell receptor on the surface. Okay. All right, antibody response, the primary response with antibody, it starts with a latent period because it takes a while to like multiply up the cells and start producing antibody. So that's the latent period where we're just starting off to make the antibody. And then we will start producing antibody, and we'll start always with IgM. And then we will class switch to IgG once we get that cranking along. Okay, but IgM is available first. So then upon our next exposure, which is our secondary or amnesiatic exposure, we will pump out primarily the IgG, which we saved up in our memory cells capabilities. So it's a much stronger, much greater response, which allows us to be exposed to things like chickenpox or measles in like real time and not have any symptoms whatsoever. You wouldn't even know you encountered it. That's how quick and reliable it is. So it's pretty cool stuff. All right. Immunity can be a natural active, which is referring to responding like to illness and getting antibodies against it. Natural passive, like what we have with antibody transfer from the mother to the child. Artificial active, dealing with vaccination. We introduced that medically. And then artificial passive, infusing somebody with some sort of gamma globulin. And that even includes things like Humira and that sort of thing. All right, vaccines. When you are developing vaccines, they can be live attenuated. That means a weakened, a live version of the virus or bacteria. Killed, self-explanatory. Subunit, just a portion of that microorganism. That's all you needed to react to it appropriately. Toxoid, that's like what we see with tetanus. Just only care about the toxin and its effects in the body. So we develop antibodies to that. Conjugated, and that's going to be associating it with taking like one organism we don't respond to very well and pairing it with one we do, and maybe really strong reaction with this one. And so we pair them together, and now that's more likely to create a strong response to the weaker one as well. So that's a useful thing, kind of pairing them together at once. Conjugating just kind of means putting things together. And then we can administer that intramuscularly, subcutaneously, intradermally, or oral nasal, of course. Remember, it's the same immune system. It's not going to respond any differently. It's still antigen response. I don't know how to describe that, but yeah. And that vaccines are kind of designed to make your immune system respond strongly. They have things, I'm sure you've heard these terms like adjuvants. An adjuvant is added to a vaccine to provoke the immune response. So there is a little bit of a difference there. Sometimes you need that extra kick to get a better response from a vaccine than you would from a natural encounter. Sometimes you need that extra kick in order to get it to be as good as a natural encounter. So, I mean, like, yeah, so whatever, but there is a little bit of difference there, but it's the same immune response in the end. You'll have the same antibodies made in the end. And then we talked about herd immunity, basically how many people have to be vaccinated in a population in order to protect the people that cannot be vaccinated. Okay. Do we want to look at this slide that I wrote on that looks like ridiculous? Let's see. I don't know. It says, apparently the computer says no. Okay, here we go. All right, let's just look over it real quick and see if it makes any sense to you guys. But you will activate the immune system. Okay, so as we're going through, this is just talking about as each thing is activated. Right. So a phagocyte here sees a PAMP. All right. And we'll go this solid line way first, right? it's going to start making cytokines and chemokines, chemicals that it secretes that induce inflammation and cause complement to be activated, right? So that's going to be a non-specific response. However, because we saw that PAMP as our phagocyte, the ones that are APCs, which isn't all phagocytes. Remember, we also have neutrophils. They are not going to be. Neutrophils are phagocytes that are not antigen-presenting cells. That's what I'm trying to say. And they are, oh, by the way, neutrophils are the most numerous cells in your blood, your white cells, of all your white cells. The neutrophils are the most numerous. That is on the test, by the way. I know it says it in like the lecture and everything, but like, and in the study guide, but I just did want to be clear about that. Okay. Antigen-presenting cells, the phagocytes that are antigen-presenting cells, the monocytes, the macrophages, the dendritic cells, the B cells. Okay, so now those guys saw the PAMP. They're part of that initial response. Okay, so they're going to go find a T helper cell, depending on whether they are presenting their antigen on MHC2 versus MHC1 due to an internal infection, and that could be from any cell, not just the special antigen-presenting ones. That TH response will lead to development of TH1 or TH2. If it was presented on MHC1, TH1 will respond. Here we go. And then that's going to lead to activation of the CD8 positive T cells. If we presented it on MHC2, we're going to activate our TH2 helper cells, and that's going to lead to production of B cells, activation of B cells and production of antibody by plasma cells. So yeah, production of antibody. So that's just kind of showing you guys a little bit of a breakdown in simple words and not all the description all at once, how all this could be associated. That's all that was meant to be. It's terrible handwriting. I apologize for that. It looked way worse on this computer than it did at work. I have a tiny screen at work that I drew it on, and I have this massive screen here at home. Oh man, and it's got that curve, so I'm like totally immersed in this terrible handwriting. Okay, so chapter 16, this is dealing with immunopathology, an important term to be aware of. I'm pretty sure it was listed as part of the vocabulary associated with this chapter, but immunopathology, that is referring to basically the study of dysregulation of the immune system, whether that is hypersensitivity or hyposensitivity, any dysregulation of the immune system. So be aware of that. Hint, hint. So my butt hurts, sorry. Hypersensitivity reactions that we talked about, type 1, type 2, type 3, and type 4. Type 1 is dealing with allergies. That is associated with IgE. That's the antibody associated with that. That IgE produced against whatever your allergen is can have an atopic reaction or an anaphylactic reaction. Atopic was going to be something that's local. That includes allergies and asthma, typical like allergies like allergic rhinitis, yeah, hay fever and that. And then anaphylactic shock, that's a reaction in the entire body all at once. Systemic. So we can be triggered by inhalants, things that you breathe in, contactants, things you touch, injectants, things that are literally poked into us, whether that's by a bee or by an actual like a vaccine, and then ingestants, things that you eat. This, you always have to have an initial sensitization for these. That means that you had to see it previously to make the IgE, and then everything after that you're responding to with that IgE. And that was associated with the mast cells, remember? And then that is also associated with production of histamine as part, one of those. I don't need you to know all the rest of all of everything that was produced, but histamine might be a good one just to be aware of. And you'll treat these with antihistamines, of course, then, or epinephrine or adrenaline. And those are typically, epinephrine is usually typically given for anaphylactic shock, not really so much for atopic reactions, but. Okay, so type 2 is dealing with, we talked mostly about the blood group interaction. So this is IgM and IgG. That leads to complement-mediated lysis of blood cells, okay? This is blood group incompatibility. We talked about the A-B-O blood groups. The A, anybody that's an A has an anti-B. Anybody that's a B has an anti-A. Anybody that's an AB doesn't have anti-anything. Anybody that's an O has anti-A and anti-B. All right. So based on that, you should be able to determine which ones would not be able to receive what blood. A can receive A and O. B can receive B and O. AB can receive A, B, AB, O, and O can receive O. That's it. All right, we talked about RH group as well, and that was the hemolytic disease of the newborn. So that was rejection of the newborn whenever an RH negative mother is having, to gestate an RH positive baby, develops antibodies against that baby's blood type, which is the RH group specifically, the positive or negative aspect of it. And then upon the second pregnancy, now that we have been sensitized to that RH group, the second pregnancy, the antibodies can attack that baby unless we block them with RhoGAM. So that was the idea with that. Can lead to death of the child if it isn't treated. I think that's everything I want to say about that. Type 3, this is immune complexes. This is whenever antibody and antigen are like all bound up with one another in these massive complexes. The big ones are cleared out. The medium-sized ones get stuck in tight spaces and in the actual basement layer of the endothelial cell layers. Yeah, and then that will trigger like reactions from basophils and neutrophils. They'll come along and say, oh, this is hanging out here, this antibodies here, this must mean we attack here. And they'll release their things to cause, inflammation and vasodilation and damage to the tissue nearby that we see as symptoms of, a lot of times autoimmune disease like lupus, right? So it's also treated with rheumatoid arthritis, but rheumatoid arthritis is way more complicated. It has a lot more stuff involved. Right, so Arthus reaction was a localized reaction of that, and serum sickness was a systemic reaction of that. It's just terms you need to know. All right, type 4 hypersensitivity. This is the delayed cell-mediated, so T cell related stuff. And then this includes the tuberculin PPD test, right? So that's why it takes 24 to 48 hours to see if you are in fact inflamed from the stuff that they injected in your skin. Poison ivy and contact dermatitis, and then as well as graft rejection. It takes some time for the body to activate the T cells associated with whatever that thing is that they're seeing as an antigen. So these also require an initial sensitization. We'll have to see something up front in order to start reacting to it. In the case of a graft, what happens is it's in there long enough that you start reacting against it. You don't need like a secondary, which is part of the initial response. So we talked about the difference between host versus graft and graft versus host. Host versus graft, that means that my lymphocytes see the donor kidney MHC1 molecule as foreign, and so we attack it, attack the cells. That's what's going on. In graft versus host, if you get a bone marrow transplant that's full of lymphocytes, those lymphocytes will recognize our cells as foreign and attack us. So the other way. The difference between these grafts: autograft from the self, like skin from another area of your body. Allograft, that's going to be from somebody else. I thought these in a different order, didn't I? Isograft is from twins. Allograft is going to be from another person. And then xenograft is going to be from the animal. Autoimmune diseases. Yeah, we talked about how these things relate to autoimmune diseases. Be aware of some of those autoimmune diseases like type 1 diabetes or, SLE or RA or whatever other ones. They're pretty common ones. If you were to see it, I'm not going to ask you to go into detail necessarily about the specifics on them, but know that they are autoimmune diseases. Forbidden clones, that's just the ones that recognize self and molecular mimicry whenever an organism mimics our cells. So a bacteria like staph might look, and this is more strep actually, though. Let's talk about that. Strep can have molecules on the surface of it that look like us to our cells. And so that's how it hides out. But if our cells were smart, and they think they're smart, and they react against it, then you can have reaction against our body as well. Due to like stuff like rheumatic fever and all that. So, but that, yeah, so seeing that as self, and then now we see self as foreign. So that's the idea with molecular mimicry. What they think was going on with type 1 diabetes and, exposure to mumps. So the immunodeficiencies that we talked about, primary, from birth, secondary, acquired. The one we talked about from birth, the bubble boy disease, SCID, SCID, severe combined immunodeficiency. And the secondary one was acquired, we talked about AIDS, but don't forget that radiation and chemotherapy can do that as well. Okay, chapter 17. Chapter 17. You guys have any questions so far? I kind of skipped over 15 and I didn't ask if you had any questions. Okay. So chapter 17, that was the diagnostic one, the short one. The short one. We talked about the different kinds of tests that you can do when you're trying to diagnose your patient. The phenotypic, the immunologic, and the genetic. Remember, phenotypic is going to be things like your biochemical tests. Immunologic, things like ELISA, and genetic, things like PCR. Steps involved in diagnosing your patient. You should be looking at the signs and symptoms of your patient first. And if this is a syndrome, what is there that you can help identify what could potentially be causing it within that syndrome? If your patient you think has meningitis, are you just going to jump to, Neisseria meningititis, which is like probably the most common and most terrible of the bacterial ones, or what? Like, how are you going to assess this? And how would you decide how you're going to test it? So we're going to collect our specimens, and we're going to look at our patient. Start with the patient as much as we can first and analyze what is actually going on with their body first. Then collect our specimens. Try to analyze directly first, looking at specimen under the microscope, not needing to grow it up first. Doing immunologic tests with it without having to do growing it up first, and genetic tests like PCR without having to grow it up first. Those things we can do without growing it up first. Hopefully, if you have enough of a sample that you're working with. If you need to culture it to get more information, you will culture it like we would with our streak plates, if that's available for that organism. Isolate it, and then do tests to identify it. What tests do we do to identify it? Typically biochemical tests like the ones we're doing in lab, right? Determine what things it can utilize and what things it can't. Usually in one of those big strips like we saw in the lecture. So you can do them all at once. So that helps you narrow it down more. Then we get our presumptive data. So it's preliminary categorization, usually the genus, kind of general idea of what this could be. And then the confirmatory data to pinpoint exactly the species. Okay, if you're cultivating these things, you need to grow it up. You would have potentially isolation media that you're going to try to isolate the organism on. You can also be using differential media. Selective media can be part of it as well. And blood cultures. All of these things are important tools in cultivating whatever this organism could be. Biochemical tests are also very useful, and use of dichotomous keys, or like in the case of those strips that we were looking at, the number that you type in is essentially just doing the dichotomous key for you, but it is the same concept. So, but yeah, so that's often based on color change of the media. PCR, polymerase chain reaction, we can multiply up a very, very small amount of stuff. Antimicrobial sensitivity, using the Kirby-Bauer method, that's one way to do it. Determine, sometimes that can help you determine what antibiotic you can use, sure, but it can also help you determine what that organism is sometimes, based on how it is susceptible to one thing or not, sometimes. So, and then we have immunologic tests. The important concept that we introduced for this was specificity versus selectivity. Sensitivity. I don't know why it must have auto-corrected. I apologize. Specificity versus, now if I have to write this out because this is going to bother me. Specificity versus sensitivity. Sorry, this does not work as well. I thought I checked over all this. I probably auto-corrected some terrible thing. I'm not that great at spelling, so I probably auto-corrected it. So specificity is dealing with, eliminating any of the false positives. So you are only binding with your antigen, and you're not going to be binding with anything else and being triggered as positive for anything else. So that's the idea with that. But you might need more of your sample. You might need more of your sample in order to be more specific. Whereas if you're being having sensitivity, then that one basically like will pick up any little amount in there. So just the tiniest amount in there, it'll pop positive. However, it might pop positive from not the right antigen as well. Not always the case. Hopefully both of them are the best as possible, but you might be sacrificing one over the other. So that's the difference between specificity versus sensitivity. In vitro serum testing. This is literally taking a blood sample from somebody, spinning it, letting it clot and spinning it down and using that serum to do tests with. We can do immunofluorescence, which is a lot like ELISA. We talked about indirect and the direct, and we can confirm any of our testing that we might have done, like the immunocolorimetric ones, like we do at home with COVID. We can confirm any of that, a lot of that stuff with Western blots. That's taking the patient's sample, running it out onto a gel, transferring it to a membrane, and then mixing it with an antibody to see if that antigen is there. It's very sensitive and very specific. So you can know that, yes, this is this exact thing. We use that to confirm. The indirect, that's going to be checking, remember, to see if that patient has the antibody. Start with a known antigen. Does the patient have an antibody? Direct. Is the antigen present? Start with a known antibody. Does the patient have the antigen? You have to have a known one or the other. You have to have either known antibody or known antigen, one or the other. Okay. All right. And then we had phage typing. And what does that mean? It is something that you guys need to know about that I don't talk about enough, I don't feel like. But phage typing is essentially bacteriophages can only infect very specific bacteria. There's bacteriophage that only infects E. coli or something. So if I mix my bacteria with, my, known this only infects E. coli phage, and I see that it's killing my bacteria, then that's E. coli. It's the only one it can be. So that's phage typing, using a known phage to see if this is the organism that it can infect. So that's that. All right, so that is it for my BS for the review. I hope I went over most of it. And I hope I answered most of your questions if you had any. I don't know. I feel like a lot of this stuff is just new stuff, so it's like new, but it's whatever. So if you guys have any questions, let me know. I'll sit here for a minute and then I'll get off of here. I just wish it wasn't windy or whatever it is outside. Really windy. Yeah, I don't mind doing this stuff. What's, an hour or two out of my life. I don't mind. All right, well, if you guys don't have any questions, those of you who have sat through all this crap, if you guys don't have any questions, then I'm gonna go ahead and log off, and I will be posting this video on, on Canvas for everybody, so. Thanks for coming, guys.